Hello, everyone. My name is Leah, and I will introduce a paper published in iMeta titled Gut Microbiome and Metabolome Characteristics of Patients with Cholesterol Gallstones Suggest the Preventive Potential of Prebiotics. Gallstones are a common disease that occurs frequently. Gallstones affect around 10% to 15% of adults, leading to significant discomfort. To date, cholecystectomy remains the most effective treatment for chronic carriers with gallbladder lithiasis. As a result, gut microbiota and its metabolites, including SCFAs, hold potential as novel therapeutic targets for gallstones. This cross-sectional study aims to comprehensively evaluate potential biomarkers in patients with gallstones and assess the effects of microbiome-targeted interventions in mice. Metabolites of amino acids, fatty acids, and nicotinic acid may affect the synthesis, transport, or metabolism of cholesterol, thereby impacting gallstone formation. Oral administration of C-glycyrrhizinolyticum can contribute to cholesterol gallstone formation. Fructooligosaccharides could serve as an inexpensive microbiota-targeted therapeutic option for cholesterol gallstones. A total of 119 subjects were recruited from Beijing Hospital. After matching for age and sex variables, differential analysis was performed. Biomarkers were identified using linear and logistic regression statistical methods. Experimental validation was conducted for candidate species, SCFAs, and prebiotics. The biliary microbiota is significantly different from the gut microbiota. Our results divided 191 samples into three distinct clusters, a C, at the genus level, characterized by gut microbiota, the crossover between gut and biliary tract microbiota, and the biliary tract microbiota. A total of 56 differential species were identified through the analysis. LEFSA analysis identified 13 key features at the species level. The AUC values of the candidate species ranged from 0.65 to 0.68. Functional enrichment analysis revealed a significant increase in the activity of lipid metabolism pathways. Differential genes were concentrated in butanoate and propanoate metabolism, fatty acid degradation, and glyoxylate and dicarboxylate metabolism. Specifically, 121 metabolites were different. Representative differential metabolites were primarily classified into amino acids, carbohydrates, hydroxycinnamic acids, pyridine carboxylic acids, lipids, pterins and derivatives, and benzenoids. Functional pathways were predominantly enriched in histidine metabolism, taurine and hypotaurine metabolism, butanoate metabolism, and alpha-linolenic acid metabolism. The candidate genera, species, and metabolites were able to distinguish CGS patients from healthy controls. Various differential metabolites were involved in alpha-linolenic acid metabolism, alanine, aspartate and glutamate metabolism, nicotinate and nicotinamide metabolism, and histidine metabolism. Summarize the identification of potential biomarkers at the species and metabolite levels, and perform experimental validation for the candidate species biomarkers. The presence of C-glycyrrhizinolyticum significantly increased gallstone formation with visible leaflet crystals or cholesterol particles observed in gallbladder. Our NaVus intervention also led to a notable increase in gallstone formation but without statistical significance. Mice gavaged with AF prosnitzii still developed gallstones. Marked liver histological damage in the C-glycyrrhizinolyticum and our NaVus groups compared to control. Sodium butyrate, sodium propionate, or sodium acetate did not eliminate cholesterol particles from the gallbladders, although they reduced leaflet crystals or stratified crystals compared to the LD group. Mice treated with FOS showed no visible leaflet crystals or cholesterol particles in their gallbladders. Grade of experimental gallstones in control mice was significantly higher than that in mice treated with NaA and FOS. The study outlined the microbiome and metabolome characteristics of patients with cholesterol gallstones, summarized the related metabolic pathways, and identified disease biomarkers. Discovered a stone-promoting bacterium, C-glycyrrhizinolyticum, which exacerbates gallstone formation in mice. Prebiotics, fructooligosaccharides, may serve as a potential microbiota-targeted preventive or therapeutic strategy for cholesterol gallstones.